Hi everyone, I am Halil İbrahim Kuru. I am a PhD student at Computer Engineering Department of Bilkent University. I am happy to present our current research. It's a deep learning framework for deep drug synergy prediction, and it's a joint work with my supervisors Ercüment Çiçek and Öznur Taştan. As you know, cancer is a heterogeneous disease and cancer cells can adapt to current situation. It's possible that the cell can develop resistance to single drug therapies. Therefore, drug combination therapies have been used for several decades due to its advantages over single drug therapy. It can reduce the drug resistance, toxicity, and also reduce the side effects. Early efforts to assess drug synergy was based on clinical experiences by doctors, but it's not possible to try all combinatorial drug space, and also trying an unknown combination may lead to vital consequences. We can measure the synergy of drug pairs with high throughput screens, but even with high throughput screens, the exhaustive testing is not possible, is not feasible due to the time and money constraints. To overcome the problems in clinical and screening experiments, scientists develop computational models by using machine learning algorithms with available information from in vitro experiments. These computational models can reduce the number of drug combinations by finding reliable synergistic drug pairs. We can evaluate the candidate drug pairs in lab experiments, and drugs that pass these steps can be approved to use in clinical studies. We can use different sets of features to represent the drugs and cell lines. For example, we can represent drugs with their chemical structures, target proteins, or pathways, and we can even use the drug interaction networks. The cell lines can be represented with protein interaction networks and their omics data, such as gene expression and mutation profiles. However, the dataset sizes get smaller when we try to use all such feature sets, and this may lower the performance of the models, especially the deep learning architectures. There are different models in the literature, but we focused on two algorithms which only rely on the chemical structure and the gene expression information. The first one is Deep Synergy, a deep learning model. The second one is the tree combo. It is an XGBoost algorithm on drug synergy prediction. We use DrugCom database, which is the largest available database of the drug synergy. It combines all the studies in the literature. The initial dataset contains around 500,000 samples. Since the DrugCom contains combinations from different studies, as you see in the table, we had to do some pre-processing, which includes feature filtering and duplicate removal. At the end, we work with nearly 300,000 samples that contains more than 3,000 drugs and 81 cell lines. This huge dataset size enables training architectures like complex architectures like ours. Name of our model is MatchMaker. Before explaining our model, I first want to show the architecture of the competitor work, Deep Synergy. They basically combine all the chemical structure features of drugs with the cell line gene expression data and build their neural network model on top of these features. But in our architecture, we use three subnetworks. Instead of combining all the features at the beginning, we first consider each drug individually. We combine chemical representation of each drug with the cell line gene expression profile, and then we fed these features into two independent subnetworks called drug-specific network. These networks give latent representations of each drug, which are conditioned on cell line gene expression. These transformed representations then concatenated and passed through the third subnetwork to predict the lowest score of given drug pairs. High synergistic scores are less likely to be noise, therefore they are more interesting for life scientists. And therefore we used a weighted mean square error to penalize high synergistic scores more. Here you see our results. The original task is a regression task, and we compared MatchMaker with other methods in terms of mean square error, Pearson, and Spearman correlations. As you can see in the left figure, MatchMaker provides the lowest error and the highest correlations. Further, we evaluate our model on classification task. In general, scientists do not focus on the lowest scores around 0, and they only care about the high magnitude scores. Therefore, we discretize our predictions as synergistic. If the drug pair has lowest score higher than 10, and antagonistic if their score is less than -10. As you can see in the right figure, the curve of the matchmaker dominates all the others and achieves the highest AUC score. Here I am showing the top predictive drug pairs that our model can accurately predict the synergism of drug pairs. Here in this table, I am showing the top 25 confidence false positive predictions of our model. Here you see the actual and the predicted lowest scores of drug pairs. On these columns, we provide synergy scores from other metrics that are also available at DrugComp. For example, in the first example, although the lowest score is highly negative, we predicted it as highly positive, and all the other metrics supports our predictions as well. Further, we check the literature and we find that these two drugs interact, and the first drug can increase the serum concentration of the other drug. Another example is this combination. This combination has been shown to prolong the survival by decreasing the tumor growth and increasing the apoptosis in GBM tumors. We also applied our algorithm on individual studies to see whether our algorithm can be generalized or not. Therefore, we used O'Neill and Almanac datasets. Again, here, Matchmaker achieves the highest correlations and the lowest error. As a conclusion, we proposed Matchmaker. It's a deep learning based model which utilizes the drug chemical structure and the cell line specific gene expression patterns to predict whether two drugs are synergistic or not. We only use the drug chemical structure as the primary feature, and we use cell line specific gene expression profile to capture the context of the experiment. As you can see in our results, Matchmaker outperforms the state-of-the-art approaches in terms of predicting lowest score. Although we train our model on cancer cell lines and the pre-trained model is biased towards the cancer, the framework can be applicable to other conditions and diseases. We can use pre-trained model and fine-tune it for other cell lines, drugs, and even other diseases as well. I would like to thank Science Academy of Turkey for their support in our research. Thank you for your attention. You can reach our paper from this QR code. Our implementation is available at this GitHub link, and also you can also access the pre-trained model. Once again, thank you.